Good evening, students. So, ಇವತ್ತು ನಾವು ಬಯೋಟೆಕ್ನಾಲಜಿ ಆ್ಯಂಡ್ ಇಟ್ಸ್ ಅಪ್ಲಿಕೇಶನ್ ಈ ಒಂದು ಚಾಪ್ಟರ್ ಅನ್ನ ಕಲ್ತ್ಕೊಳ್ಳಿಕ್ಕಿದ್ದೇವೆ. ಐ ಹೋಪ್ ಆಲ್ ಆರ್ ರೆಡಿ ಟು ಲಿಸನ್ ದ ಕ್ಲಾಸ್. ಎಲ್ಲರೂ ರೆಡಿ ಇದ್ದೀರಾ ಅಂತ ಅನ್ಕೊಳ್ತಾ ಇದ್ದೇನೆ. ಸೊ ಆಲ್ರೆಡಿ ಲಾಸ್ಟ್ ಚಾಪ್ಟರ್ ಅಲ್ಲಿ ನಾವು ಬಯೋಟೆಕ್ನಾಲಜಿ, ಸೊ recombinant DNA technology, especially the steps involved in the recombinant DNA technology. So ಇದನ್ನೆಲ್ಲ ಕಲ್ತ್ಕೊಂಡಿದ್ವಿ ಅಲ್ವಾ. ಸೊ now its application. So in which field we can use the biotechnology? Okay. So ಯಾವ ಫೀಲ್ಡ್ ಅಲ್ಲಿ ನಾವು ಬಯೋಟೆಕ್ನಾಲಜಿನ ಯೂಸ್ ಮಾಡಬಹುದು? ಸೊ how we can improve the lifestyle by using the biotechnology. So and its application, so that we are studying the application of the ಬಯೋಟೆಕ್ನಾಲಜಿ. ನೋಡಿ, ಬಯೋಟೆಕ್ನಾಲಜಿಯನ್ನ ನಾವು ಯಾವ ಫೀಲ್ಡ್ ಅಲ್ಲಿ ಅಪ್ಲೈ ಮಾಡಬಹುದು ಅಂದ್ರೆ ಫಾರ್ಮಾಸ್ಯೂಟಿಕಲ್ಸ್ ಮೀನ್ಸ್ ಟು ಪ್ರಿಪೇರ್ ದ ಸಮ್ ಮೆಡಿಸಿನ್ಸ್. ಓಕೆ, ದೆನ್ ಥೆರಾಪ್ಟಿಕ್ಸ್ ಅಂಡ್ ಡಯಾಗ್ನೋಸ್ಟಿಕ್ಸ್ ಅಲ್ಲೂ ಕೂಡ ಯೂಸ್ ಮಾಡಬಹುದು. ಟು ಡಯಾಗ್ನೋಸ್ ಸಮ್ ಡಿಸೀಸಸ್, ಜೆನೆಟಿಕಲಿ ಮಾಡಿಫೈಡ್ ಕ್ರಾಪ್ಸ್. ಸೊ ಕ್ರಾಪ್ಸ್ ಜೆನೆಟಿಕಲಿ ಮಾಡಿಫೈ ಮಾಡಿ ಆ ಒಂದು ಪ್ಲಾಂಟ್ಸ್ ಅನ್ನ ಈಲ್ಡ್ ಇರಬಹುದು ಅಥವಾ ಡಿಸೀಸ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಕೆಪಾಸಿಟಿ ಇರಬಹುದು, ಈ ರೀತಿಯಾದಂಥ ಕೆಲವೊಂದು ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಅನ್ನ ಇಂಪ್ರೂವ್ ಮಾಡಲಿಕ್ಕೆ ವಿ ಕ್ಯಾನ್ ಯೂಸ್ ದ ಬಯೋಟೆಕ್ನಾಲಜಿ ಟು ಗೆಟ್ ದ ಜೆನೆಟಿಕಲಿ ಮಾಡಿಫೈಡ್ ಕ್ರಾಪ್ಸ್. ದೆನ್ ಪ್ರೊಸೆಸ್ಡ್ ಫುಡ್ಸ್, ದೆನ್ ಬಯೋರೆಮಿಡಿಯೇಷನ್, ದೆನ್ ವೇಸ್ಟ್ ಟ್ರೀಟ್ಮೆಂಟ್, ಎನರ್ಜಿ ಪ್ರೊಡಕ್ಷನ್. ಸೊ ಈ ಎಲ್ಲ ಫೀಲ್ಡ್ ಅಲ್ಲಿ ನಾವು ಬಯೋಟೆಕ್ನಾಲಜಿ ಅಪ್ಲಿಕೇಶನ್ ಅನ್ನ ನೋಡ್ಬೋದು. ಓಕೆ, ಮೇನ್ಲಿ ಪ್ರೊಡಕ್ಷನ್ ಆಫ್ ದಿ ಮೆಡಿಸಿನ್ಸ್ ಅಂಡ್ ಡಯಾಗ್ನೋಸಿಸ್ ಆಫ್ ಡಯಾಗ್ನೋಸ್ಟಿಕ್ಸ್ ಲೈಕ್ ಡಿಸೀಸಸ್ ಅಂಡ್ ಫುಡ್ ಪ್ರೊಡಕ್ಷನ್, ವೇಸ್ಟ್ ಟ್ರೀಟ್ಮೆಂಟ್. ಸೊ ಈ ಎಲ್ಲ ಫೀಲ್ಡ್ ಅಲ್ಲಿ ನಾವು ಬಯೋಟೆಕ್ನಾಲಜಿ ಅಪ್ಲಿಕೇಶನ್ ಅನ್ನ ನೋಡ್ಬೋದು ಅಲ್ವಾ. ದೆನ್ so three critical research areas of biotechnology. So there are mainly three critical research areas. So the first critical research area is provide the best catalyst in the form of improved organism, usually a microbe or pure enzyme. So this is the first critical research area of biotechnology. Then create the optimal condition through the engineering for a catalyst to act, to create the optimal conditions that the catalyst to act. So that is the second critical research area. Then third one is the downstream processing technologies to purify the protein and organic compound. So three important so these are the three important critical research areas of the biotechnology. Then application of the biotechnology in agriculture field. Okay, so very important. Let us start with the application in agriculture. So agriculture ಅಲ್ಲಿ ಯಾವ ಪರ್ಪಸ್ಗೆ ಯಾವ ರೀತಿಯಾಗಿ ನಾವು ಯೂಸ್ ಮಾಡಬಹುದು ಅಂತ. The first thing is to increase the food production, agrochemical based agriculture, agro and chemical based agriculture, where the chemicals, pesticides and fertilizers are used. Organic agriculture, where the organic manure is used. And genetically engineered crop based agriculture. So ಈ ಮೂರು ಡಿಫ್ರೆಂಟ್ ವೆರೈಟಿ ಆಗಿರುವಂತಹ ಒಂದು ಅಗ್ರಿಕಲ್ಚರ್ ಫೀಲ್ಡ್ ಅಲ್ಲಿ ನಾವು ಬಯೋಟೆಕ್ನಾಲಜಿಯನ್ನು ಅಪ್ಲೈ ಮಾಡಬಹುದು. ಸೊ ವೈ ಬಯೋಟೆಕ್ನಾಲಜಿ ಇಸ್ ಯೂಸ್ಡ್ ಇನ್ ಅಗ್ರಿಕಲ್ಚರ್ ಅಲ್ಲಿ ಬಯೋಟೆಕ್ನಾಲಜಿಯನ್ನು ಯಾಕೆ ಯೂಸ್ ಮಾಡ್ತಾರೆ? ಇನ್ಕ್ರೀಸ್ ದ ಫುಡ್ ಪ್ರೊಡಕ್ಷನ್ ಅಂಡ್ ದ ಫುಡ್ ಕ್ವಾಲಿಟಿ. ದಟ್ ಈಸ್ ದ ಮೇನ್ ರೀಸನ್ ಆಫ್ ದಿ application of biotechnology in agriculture. The first one is agrochemical based agriculture to increase the food production, and organic agriculture and genetically engineered crop based agriculture. Then genetically modified organisms are the plants, bacteria, fungi and the animals whose genes are altered by the ಮ್ಯಾನಿಪುಲೇಷನ್. ಸೊ ಕೆಲವೊಂದು ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ, ಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ ಅಥವಾ ಅನಿಮಲ್ಸ್ ಇರಲಿ, ಏನ್ ಮಾಡಿರ್ತಾರೆ, ಅದರ ಜೀನ್ಸ್ ಅನ್ನ ಚೇಂಜ್ ಮಾಡಿರ್ತಾರೆ. ಒರಿಜಿನಲ್ ಜೀನ್ ಅನ್ನ ತೆಗೆದು ಬೇರೆ ಒಂದು ಡಿಸೈರ್ಡ್ ಜೀನ್ ಅನ್ನ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿರ್ತಾರೆ, ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಜೆನೆಟಿಕಲಿ ಮಾಡಿಫೈಡ್ ಆರ್ಗಾನಿಸಮ್ಸ್. ದೇ ಆರ್ ಕಾಲ್ಡ್ ಆಸ್ GMO. GMO means genetically modified organisms. Okay. Then ಅಡ್ವಾಂಟೇಜಸ್ ಆಫ್ ಜೆನೆಟಿಕ್ ಮಾಡಿಫಿಕೇಶನ್ ಇನ್ ಪ್ಲಾಂಟ್. ಸೊ ಯಾವುದೋ ಒಂದು ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಅದರ ಒರಿಜಿನಲ್ ಜೀನ್ ಅನ್ನ ರಿಮೂವ್ ಮಾಡಿ, ಸೊ ಇಫ್ ದೇ ಹ್ಯಾವ್ ಇಂಟ್ರೊಡ್ಯೂಸ್ ದ desired gene. So why, what is the advantage may we get by using this modification? So it makes the crops more tolerant to abiotic stress like cold, drought, salt and heat. So the plants, they may tolerate the high cold or drought condition or salt. So during heat also, so to tolerate these all abiotic stress. So that is the one advantage of the genetic modification. Then pest resistant crops reduce the use of chemical pesticide. So by using the biotechnology, we can produce the pest resistant plant. So genetically they are producing some chemicals that is not suitable for the pest, that may kill the pest. So we can reduce the use of the pesticides. So that is the pest resistant crops. Then it helps to reduce the post harvest loss. ಸೊ ಕೆಲವೊಂದ್ಸಲ ಏನಾಗತ್ತೆ, ಆ ಹಾರ್ವೆಸ್ಟಿಂಗ್ ಆದ್ಮೇಲೆ ಅದೇನಾಗತ್ತೆ ಕೆಲವೊಂದ್ಸಲ, ಸೊ ದಟ್ ಮೇ ಬಿಕಮ್ loss. Okay, so that is called as, so it helps to reduce the post harvest losses. It increases the efficiency of mineral usage by plants. It prevents the early exhaustion of soil fertility. Then it enhances the nutritional value. So sometimes the nutritional value also increased by using the genetic modification. So ಅದರಲ್ಲಿರುವಂಥ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಕ್ವಾಲಿಟಿನನ್ನ ಇನ್ಕ್ರೀಸ್ ಮಾಡ್ಬೋದು. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಗೋಲ್ಡನ್ ರೈಸ್ ವೇರ್ ದ ವಿಟಮಿನ್ ಎ ಈಸ್ rich, that is the vitamin A rich rice. Then genetically modified organisms is used to create tailor-made plants to supply the alternative resources such as starch, fuel and pharmaceuticals to industries. So we can use the biotechnology, so genetical modification in the agriculture for these all purpose. Okay, very important. Next one is, what is this pest resistant plants? Pest resistant plants act as biopesticide, means they are able to control the pest themselves. Okay, no pesticide is needed to control the pesticides. It reduces the need for insecticides. For example, bt cotton, bt corn, rice, tomato, potato, soybean, etc. So what is this bt cotton? How these plants, pest resistant plants are developed? Okay, so let us see about the bt cotton. What is this bt cotton and how this plant is developed the pest resistance capacity? Okay, let us see about the bt cotton. So some strains of bacillus thuringiensis have proteins that kill insects like coleopterans and beetles, lepidopterans and tobacco budworms. So some worms, so the some strains of bacteria, so this bt bacillus thuringiensis bacteria, that is able to kill these worms. Okay, so this Bacillus thuringiensis forms a toxic insecticidal protein, so that is called as Bt toxin crystals during a particular phase of their growth. It does not kill the bacillus as it exists as an inactive protoxin. So ಈ Bacillus thuringiensis bacteria ಏನಿದೆ ಅಲ್ವಾ, so that produces some toxin that is called as Bt toxin. Okay, during the particular stage of their growth, and which is in an inactive form, that is called as prototoxin. That is not, that does not kill the bacillus, that does not kill the bacteria, because that is in the inactive form. Okay, and then When an insect ingests the inactive toxin, it is converted into active toxin due to the alkaline pH of gut which solubilize the crystals. So ಇಲ್ಲೇನಾಗುತ್ತೆ ಅಂದ್ರೆ ಈ BT toxin ಅನ್ನ so when these worms they consume this BT toxin become active in the gut of their in the gut and due to the alkaline pH, and toxins binds to the surface of the midgut and creates the pores, and it causes cell swelling and lysis and death of the insect. So like this it causes the death of the insect. Okay, so this is how the BT toxin functions. BT toxin ಅನ್ನ ಇರುವಂತಹ ಒಂದು ಪ್ಲಾಂಟ್ ಅನ್ನ ಅಥವಾ BT ಟಾಕ್ಸಿನ್ ಏನಾಗಿರುತ್ತೆ ಇನ್ಆಕ್ಟಿವ್ ಫಾರ್ಮ್ ಅಲ್ಲಿ ಇರುತ್ತೆ. ಸೊ ಇಟ್ ವಿಲ್ ಗೆಟ್ ಕನ್ವರ್ಟ್ ಇನ್ ದ ಗಟ್ ಆಫ್ ದಿ ವರ್ಮ್ ಡ್ಯೂ ಟು ದ ಅಲ್ಕಲೈನ್ pH. ಸೊ ಆಕ್ಟಿವ್ ಆದಾಗ ಏನ್ ಮಾಡುತ್ತೆ ದಟ್ ಕಾಸಸ್ ದ ಲೈಸಿಸ್, break down of the cells and death of the insect. Okay, so that causes the cell death. Then how these plants are developed? BT cotton plant is developed, okay, by using the biotechnology. BT toxin genes were isolated from the Bacillus thuringiensis and incorporated into crop plants such as cotton. Okay, a Bacillus thuringiensis BT toxin anna produce ಮಾಡುವಂತ genes ಅನ್ನ they have extracted it from the Bacillus thuringiensis bacteria. Okay, the genes that is able to produce this toxin that is isolated and that is incorporated into a crop plant such as cotton. So this BT Bacillus thuringiensis gene that is incorporated into the plant ಗೆ ಅದನ್ನ incorporate ಮಾಡಿದಾಗ so the toxin is coded by gene CRY1AC and named as CRY gene. Okay, so that BT toxin producing gene that is called as CRY gene. Proteins encoded by genes are CRY1AC, CRY2AB and controls the cotton bollworms, and that CRY1AB controls the corn borer. So illi genes. So they are going to produce the different proteins, so that controls the different bollworms. Okay, so when the genes are incorporated, BT genes are incorporated into the plant is able to develop the some BT toxin. So when the bollworm when they start to feed on this plant, the protein that will get activated in their body and that cause the death of the bollworm. Okay, so this is how the BT cotton plant is developed. So why Bacillus thuringiensis gene is introduced? Because to reduce the use of the pesticides. Okay, so like this the BT cotton plants are developed. So this is the first application, that is the Bacillus thuringiensis. Okay, then second one is the pest resistant plant, that is the nematode resistance in tobacco plants. Here the nematode, so that is the roundworm, so Melodygine incognita also it is called, infects the roots of the tobacco plants and causing the reduction of the yield. Okay, so this Melodygine nematode that is causing the that affects the roots of the plant and that reduces the yield. This is also we can control by using the method RNA interference. What is this RNA interference? So it is the cellular defense in eukaryotes. It prevents the translation of the specific mRNA due to the complementary double strand RNA molecule. So ಹಾಗಾದ್ರೆ ಈ ಒಂದು ನೆಮಟೋಡ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಪ್ಲಾಂಟ್ ಅನ್ನ ಹೇಗೆ ಡೆವಲಪ್ ಮಾಡ್ತಾರೆ? ಬೈ ಯೂಸಿಂಗ್ ದ ಬಯೋಟೆಕ್ನಾಲಜಿ. ಓಕೆ, ಸೊ ಹಿಯರ್ the plant, so just imagine this is the tobacco plant. Okay, so the plant is incorporated with the gene. So that is the nematode specific gene. Okay. So using the agrobacterium vector, nematode specific genes, that is DNA, is introduced into the host. So just imagine this is the nematode specific gene that is introduced into the plant. Now nematode specific gene is present in this plant. Okay. Now this nematode resistance gene that produces the, you know, that DNA that produces the RNA. Okay. So that has two strands. The RNA has the two strands. Commonly, you know that RNA that has the single strand, but it has the dsRNA, means double strand RNA. So that is called as sense and antisense strand. So the RNAs are complementarity double strand, so they form the double strand RNA. So ಕಾಮನ್ ಆಗಿ ಏನಾಯ್ತು, when the nematode entered to this plant, so nematode entry ಆದಾಗ ಏನಾಗತ್ತೆ, they they form the double strand RNA. It initiates the RNA interference and silence the specific mRNA of the nematode. ಈ nematode ಏನ್ ಮಾಡುತ್ತೆ ಅಂದ್ರೆ, so it will start to cause the disease, but it produces the, to cause the disease, it produces the RNA, you know that RNA that is able to produce the protein, and that protein that cause the disease ಅಲ್ವಾ. ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ ಏನಾಗತ್ತೆ, DNA, so that produces the RNA that produces the protein is responsible for the characters, that is the disease. So already the similar DNA is introduced to into the plant, and that is also produce the double strand RNA. So that the double strand RNA that binds this RNA and which prevent the translation of this RNA, and the protein is not produced. If the protein is not produced means, so there is no disease. Okay. So this is called as RNA interference. So the already the nematode specific gene is that is introduced into the plant, and that produces the double strand RNA. Okay. So when the nematode that produces the RNA, okay, so to cause the disease, when the nematode enter the tobacco plant and which introduce its DNA into the plant, and that DNA that produces the RNA, but this RNA that is interfere, that expression of RNA is interfered by the double strand RNA, okay, by producing the complex. The protein is not produced. Protein is not produced means, so there is no disease development. Okay. So it is called as RNA interference. So that is the silencing of the specific mRNA of nematode. So thus the parasite cannot survive in the transgenic host expressing the specific interfering RNA. So ಆ RNA produce ಆದಾಗ ಏನಾಗುತ್ತೆ, so which is producing the RNA interference, so that is not able to live in the host plant. Okay. So this is how RNA interference that reduces the crop, that reduces the nematodes. Okay. Then second one is application in medicine. So in medicinal field, how this biotechnology, so that is used. Recombinant DNA technology help for the mass production of safe and more effective therapeutic drugs. Okay. The products from non-human sources induce unwanted immunological responses, but recombinant, so these products, so they are not have the such products. Okay, this recombinant medicines, they do not have such type of the effect. So application in the medicine field, so the first one is genetically engineered insulin, gene therapy and molecular diagnosis we are studying. Okay. Then genetically engineered insulin. So you know that insulin in our body, that is produced by the pancreatic gland and which maintains the blood sugar level, right? So if the person is suffering from the diabetes mellitus means, so the pancreas that is not able to form the enough insulin in their body. Okay, so insulin from pancreas of the animals like cattle and pigs causes allergy or other types of reaction to the foreign protein. So now it's possible to produce the human insulin using the bacteria. So now using the biotechnology, we are producing the human insulin also. Okay, so how it is going to produce? Insulin consists of two short polypeptide chains, so that is chain A and chain B, that are linked by disulfide bond. So you can see the insulin A and insulin B, and that is bonded by disulfide bond. In mammals, insulin is synthesized as a prohormone. It needs processing to become mature and functional hormone. So ಇಲ್ಲಿ mammals ಅಲ್ಲಿ ಏನಾಗಿರುತ್ತೆ, so the insulin that has that should undergo maturation, uh, it should undergo some process to become a mature insulin. Then so this insulin that has A chain, B chain along with the stretch C chain. Okay, free C peptide chain is also present in case of the prohormone. Okay, so removal of this C chain, so that leads to the formation of the mature insulin. Mature insulin has only A and B strand. Okay, mature insulin ಅಲ್ಲಿ A ಮತ್ತೆ B strand ಮಾತ್ರ ಇರುತ್ತೆ. So let us see the production of the insulin.
Recombinant DNA technology can be used in the production of human insulin. Now, let's take a look at the production process. First, DNA encoding human insulin is obtained from a human pancreatic cell. Then, a restriction enzyme is used to cut the DNA. Meanwhile, a plasmid with antibiotic resistance genes is obtained from a bacterium. The plasmid is cut open using the same restriction enzyme. When the DNA fragments and the open plasmids are mixed together, some of the plasmids bind with the DNA fragments, and with the help of DNA ligase, they are joined together, forming recombinant plasmids. Note that some open plasmids do not pick up the DNA fragments and join by themselves. They are called non-recombinant plasmids. The plasmids are then mixed with the bacteria E. coli. This is to introduce the plasmids into the host cells. However, only some bacteria actually pick up the plasmids. These bacteria are said to be transformed, and only some of them have the recombinant plasmid. The remaining transformed bacteria have the non-recombinant plasmid. Meanwhile, some bacteria do not pick up any plasmids at all. In this example, the plasmid we use carry a resistance gene for the antibiotic ampicillin. So, to select the transformed bacteria, We first culture the bacteria on an agar plate containing ampicillin. The ampicillin resistance gene in the plasmid enables the transformed bacteria to survive. They then divide and form colonies. The bacteria that didn't pick up any plasmid cannot survive. After that, we transfer a portion of bacteria from each colony to a second agar plate containing another antibiotic tetracycline. As the transformed bacteria with a non-recombinant plasmid carry an intact tetracycline resistance gene, they survive and divide to form colonies. On the other hand, in the transformed bacteria with a recombinant plasmid, the DNA fragment inserted made the tetracycline resistance gene non-functional. Therefore, the bacteria cannot survive on the plate. From the results of these two agar plates, we can identify the colony formed by the transformed bacteria with a recombinant plasmid, and select these bacteria for further processing. The transformed bacteria with a recombinant plasmid are then cultured in fermenters. In each bacterium, the recombinant plasmid replicates independently of the bacterial chromosome. When the bacterium divides, the plasmid is also copied to the daughter cells. As a result, many copies of the DNA encoding human insulin are produced. Finally, gene expression is induced in the bacteria to produce polypeptides. The polypeptides are extracted from the bacteria and processed into functional human insulin.
Yes, so what we have seen in the video, that is the detailed method of how the insulin is produced. Okay. Then in the year 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B human chain. So which produces the A insulin chain and the B insulin chain. They were introduced in a plasmid of E. coli to produce the insulin chains, and chains A and B were combined by creating the disulfide bonds to form the human insulin. So to produce the human insulin, so these were introduced into the plasmids of E. coli, and A and B were joined by the disulfide bond. Okay, so like this the human insulin is produced. So that insulin, so that is used by the diabetic patients. Okay, so how this genetically engineered insulin is produced using the biotechnology. Okay, so that is the second thing. And the gene therapy means correcting. Okay, so gene therapy means correcting the defected gene. Okay, so it is a method to correct the gene defect in a child or embryo. So in the embryonic state only, the correction of the gene, so defected gene, so that is called as the gene therapy. The genes are inserted into a person's cells and tissues to treat a hereditary disease, to compensate for the non-functional gene. Sometimes the non-functional gene that may cause the disease. Okay, so that genes are removed and functional genes are introduced in the gene therapy. Then the first gene therapy was given to a 4-year-old girl. So that is having the adenosine, she's having the adenosine deaminase, that is ADA deficiency. So what is this deficiency cause? So this deficiency, this enzyme that is for the immune system function. Immune system function means to protect our body from the some foreign bacterial viral agents. Okay, so this is very important enzyme, that is the adenosine deaminase. Treatment by bone marrow transplantation or enzyme replacement therapy, or that is the injection of ADA, is not completely curative. So this disease, so that also can be cured by bone marrow transplantation. So ನಮ್ಮ ಬೋನ್ ಮ್ಯಾರೋನ where the new body cells are produced, right? ಬೋನ್ ಮ್ಯಾರೋದಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಬಾಡಿ ಸೆಲ್ಸ್, ರೆಡ್ ಬ್ಲಡ್ ಸೆಲ್ಸ್, WBC ಎಲ್ಲ ಪ್ರೊಡಕ್ಷನ್ ಆಗತ್ತೆ ಅಲ್ವಾ. ಸೊ ಆ ಬೋನ್ ಮ್ಯಾರೋ ಟ್ರಾನ್ಸ್ಪ್ಲಾಂಟೇಶನ್ ಅನ್ನ ಮಾಡಿ, ಸೊ ಆರ್ enzyme replacement where the directly ADA enzyme is injected. So these both methods are not completely curative method, and she is the first girl who treated with this um, gene therapy. Okay, then ಹಾಗಾದ್ರೆ ಜೀನ್ ಥೆರಪಿಯನ್ನ ಹೇಗೆ ಪರ್ಫಾರ್ಮ್ ಮಾಡಿದ್ರು ಅಲ್ವಾ. ಸೊ ಕಲೆಕ್ಟ್ ದ ಲಿಂಪೋಸೈಟ್ಸ್ ಅನ್ನ ಕಲೆಕ್ಟ್ ಮಾಡಿ ಫ್ರಮ್ ದಿ ಪೇಷಂಟ್ಸ್ ಬ್ಲಡ್ ಅಂಡ್ ಗ್ರೋ ಇನ್ ಅ ಕಲ್ಚರ್. ಸೊ ಸೊ ದೀಸ್ ಆರ್ ದಿ lymphocytes. So they are growing in a culture. Introduce a functional ADA, so that is the functional DNA into a lymphocytes using the retroviral vector. So here you can see human ADA means the corrected gene is added to the, so this lymphocytes by using the vector. This should be periodically repeated as lymphocytes are not immortal, non-immortal. Okay. So ಈ lymphocytes ಅನ್ನ ಏನ್ ಮಾಡಿದ್ರು, so the corrected, which is having the corrected gene, so that have been multiplied and introduced into the person's body. So the lymphocytes which is having the corrected ADA gene that have been introduced into the person's body. So ಆದರೆ lymphocytes ಏನಾಯ್ತು, they may die. The lymphocytes which are having the corrected ADA gene that may die. So this is not this is the periodically repeated process. But if the ADA gene from the marrow cells is introduced into a cell at early embryonic stage, if this process could be done in early embryonic stage, then that will be the permanent cure. Otherwise the repeatedly this process should be done. Okay, so that is how the gene therapy will be worked in. Okay, so T lymphocytes are taken out, and these T lymphocytes, so they are introduced by the functional normal ADA producing DNA, and that is multiplied and introduced into the body. Okay, so that is how the gene therapy is done. Then molecular diagnosis to detect some diseases. So here conventional methods, serum and urine analysis are not suitable for the early diagnosis of the disease. Commonly we use the serum and urine analysis to detect some disease. It is possible by the technique like recombinant DNA technology, PCR and ELISA. So these techniques are used to detect some diseases. Okay, PCR, ELISA. So these are the technologies that is used to detect some diseases. Then what is this PCR? Presence of a pathogen is normally suspected only based on the symptoms. So ಯಾವುದೇ ಒಂದು ಡಿಸೀಸ್ ಕಾಸ್ ಆಗಿದೆ ಅಂದರೆ ವಿ ಕ್ಯಾನ್ ಅಬ್ಸರ್ವ್ ದ ಸಿಂಪ್ಟಮ್ಸ್, ಬಟ್ ವೆನ್ ವಿ ಅಬ್ಸರ್ವ್ ದ ಸಿಂಪ್ಟಮ್ಸ್ ಆಲ್ರೆಡಿ ದ ಪ್ಯಾಥೋಜನ್ is multiplied or that number is high in our body. Okay, but however, if the low concentration of bacteria or virus can be detected by amplification of their nucleic acid by PCR. So just imagine, so you are suspected of some disease. Okay, so if you are suffering from any disease, if the symptom is already there, means already more number of bacteria or viruses are present in your body. So sometimes symptoms are not there, but only low number of virus and bacteria is present in our body means, so you can test that disease, test that presence by multiplying their nucleic acid by using the PCR technique, that is polymerase chain reaction. Okay, then what is the uses of PCR to detect the HIV in the suspected AIDS patients? So AIDS suspected, so symptoms irodilla, only low virus particle iರುತ್ತೆ, ಆ viral particles ಅನ್ನ PCR through multiply ಮಾಡಿ we can detect the disease. To detect the mutation in genes in the cancerous patients, to identify many other genetic disorders. Okay, so that is the uses of the PCR. Then a single stranded DNA or RNA tagged with radioactive molecule is hybridized. Complementary DNA in a clone cells is followed by the detection using autoradiography. Okay, the clone having mutated gene will not appear on the photographic film because probe will not have complementarity with the muted gene. So this is the another method that detect the presence of the some other foreign DNA molecules. Okay, so ELISA. So what is this ELISA? Enzyme linked immunosorbent assay. ELISA is based on the principle of antigen antibody interaction. So this ELISA that is used to detect the AIDS. Okay, so how this ELISA is used? ELISA is based on principle of antigen antibody interaction. So what is this antigen antibody interaction? Infection by pathogen can be detected by the presence of antigens like proteins, glycoproteins, or by detecting the antibody synthesized against the pathogen. ಸೊ ಇಲ್ಲಿ ಏನಾಗಿರುತ್ತೆ, ಏಡ್ಸ್ ಅಟ್ಯಾಕ್ ಆದಾಗ ಸೊ ಇನ್ ಅವರ್ ಬಾಡಿ ಸಮ್ ಆಂಟಿಬಾಡೀಸ್ ಆರ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್ for that pathogen. So by detecting that antibodies we can detect the some diseases. Okay, by detecting which type of antibodies are produced, so we can observe the, we can see the which disease is caused. Okay, then transgenic animals. So what are these transgenic animals? Transgenic animals means which have the manipulated DNA. Okay, so instead of their original DNA molecule, they have altered or some foreign gene in their body. So they are called as transgenic animals. For example, transgenic rats, rabbits, pigs, sheep, cows. So these all are the example for the transgenic animal. So which has the some extra DNA in their body, foreign DNA introduced in their body. Okay. Then what is the benefits of these transgenic animals? What are the uses to produce these transgenic animals? To study the regulation of genes on normal physiology and development, to study the contribution of genes in the development of disease, biological products, vaccine safety and testing, chemical safety testing. So these all are the benefits of the transgenic animals. So we'll study about this in detail. Okay, the first one is to study regulation of genes on normal physiology and the development. So what is the meaning of this? So study of complex factors such as insulin and like growth factor. So when you introduce the some foreign gene, how they are working in the, how they are contributing for the development and normal physiology of that animal. So genes that alter formation of this factor are introduced and biological effects are studied. ಸೊ ಕೆಲವೊಂದು ಜೀನ್ಸ್ ಅನ್ನ ಅನಿಮಲ್ಸ್ ಗೆ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿ ಸೊ ಅದರ ಇಫೆಕ್ಟ್ಸ್ ಅನ್ನ ಏನ್ ಮಾಡ್ತಾರೆ, ಸ್ಟಡಿ ಮಾಡ್ತಾರೆ. ಸೊ ದಟ್ ಈಸ್ ಫಾರ್ ದಿ ಟು ಸ್ಟಡಿ ದ ರೆಗ್ಯುಲೇಷನ್ ಆಫ್ ಜೀನ್ಸ್ ಆನ್ ನಾರ್ಮಲ್ ಫಿಸಿಯಾಲಜಿ ಅಂಡ್ ಡೆವಲಪ್ಮೆಂಟ್, ಹೌ ಇಟ್ ಈಸ್ contributing for the development and what is the effect of this gene. So this gives information about the biological role of the factor in the body. So like that we can study about that genes in the organism's body. Okay, that is the first benefit. Second one is to study the contribution of genes in the development of a disease, how these genes are going to contribute for the development of the disease. For example, transgenic models help to investigate the new treatments for the human disease. So for example, the particular gene that is introduced into the animals and that develop the some disease. Okay, so this how this disease can be cured, that can be studied by using these transgenic animals. For example, the diseases like cancer, cystic fibrosis, rheumatoid arthritis and Alzheimer's. So these all many other diseases also can be studied by using these genes. Okay, to study the contribution of the genes. So how these genes are responsible for the development of the disease, how it can be cured. Okay, so which gene is causing the disease and how we can overcome this disease, how we can going to cure this disease. So that effect we can study. ಸೊ ಬಯಾಲಜಿಕಲ್ ಪ್ರಾಡಕ್ಟ್ಸ್ ಅಂದ್ರೆ ಕೆಲವೊಂದು ಇಂಪಾರ್ಟೆಂಟ್ ಎಸೆನ್ಷಿಯಲ್ ಪ್ರಾಡಕ್ಟ್ಸ್ ಅನ್ನ ನಾವು ಆಬ್ಟೈನ್ ಮಾಡಬಹುದು. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, transgenic animals are used to produce the biological products by introducing the genes which codes for particular product, that is human protein, that is alpha antitrypsin used to treat the emphysema. ಸೊ ಈಗ ಒಂದು ಜೀನ್ಸ್ ಅನ್ನ ಒಂದು ಆರ್ಗ್ಯಾನಿಸಮ್ ಅಲ್ಲಿ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ದಾರೆ. ಸೊ ದಟ್ ಈಸ್ ಗೋಯಿಂಗ್ ಟು produce one protein, that is called human protein, that is called as the alpha antitrypsin. So this alpha antitrypsin that is used to treat the emphysema. Okay, so products for treatment of phenylketonuria and cystic fibrosis, so these all are used. So some biological products obtained from that gene are used to treat the disease. Okay, ಈಗ ಒಂದು ಜೀನ್ ಅನ್ನ ಆರ್ಗಾನಿಸಮ್ ಅಲ್ಲಿ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ದಾರೆ. ಸೊ ದಟ್ ಜೀನ್ ಪ್ರೊಡ್ಯೂಸಸ್ ದ ಪ್ರೋಟೀನ್ಸ್ ಅಂಡ್ ದಟ್ ಪ್ರೋಟೀನ್ that is used to cure some disease. Okay, so that is the biological products. Then so one more example to obtain the biological product. In the year 1997, Rosie, so that is the first transgenic cow, okay, produced a human protein enriched milk that contained the 2.4 grams per liter. So that is also transgenic animal. ಒಂದು ಫಾರಿನ್ ಡಿಸೈರ್ಡ್ ಜೀನ್ ಅನ್ನ ಈ ಆರ್ಗಾನಿಸಮ್ ಅಲ್ಲಿ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ರು and that produced the milk which contain more protein, that is 2.4 gram per protein. ಸೊ ಅಷ್ಟು ಪ್ರೋಟೀನ್ ಇರುವಂತ ಮಿಲ್ಕ್ ಅನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿತ್ತು. ಇಟ್ ಕಂಟೈನ್ಸ್ ಹ್ಯೂಮನ್ ಆಲ್ಫಾ ಲ್ಯಾಕ್ಟಲ್ಬುಮಿನ್. ಇಟ್ ಈಸ್ ನ್ಯೂಟ್ರಿಷನಲಿ ಮೋರ್ ಬ್ಯಾಲೆನ್ಸ್ಡ್ ಪ್ರಾಡಕ್ಟ್ ಫಾರ್ ಹ್ಯೂಮನ್ ಬೇಬೀಸ್ ದ್ಯಾನ್ ದ ನ್ಯಾಚುರಲ್ ಕೌ ಮಿಲ್ಕ್. ಸೊ ನೋಡಿ, ಈ ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಕೌ ಯಾವ ಒಂದು ಪ್ರೋಟೀನ್ ಅನ್ನ ಪ್ರೊಡಕ್ಷನ್ ಮಾಡಿತ್ತು, ಆಲ್ಫಾ ಲ್ಯಾಕ್ಟಲ್ಬುಮಿನ್. ಸೊ ದಟ್ ಈಸ್ ವೆರಿ ಎಸೆನ್ಷಿಯಲ್ ಫಾರ್ ದಿ ಹ್ಯೂಮನ್ ಬೇಬೀಸ್. ಓಕೆ, ಸೊ ಈ ರೀತಿಯಾಗಿ ಬಯಾಲಜಿಕಲ್ ಪ್ರಾಡಕ್ಟ್ ಅನ್ನ ನಾವು ಆಬ್ಟೈನ್ ಮಾಡಬಹುದು ಬೈ ಯೂಸಿಂಗ್ ದ ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಅನಿಮಲ್ಸ್. ವ್ಯಾಕ್ಸಿನ್ ಸೇಫ್ಟಿ ಟೆಸ್ಟಿಂಗ್. ಸೊ ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಮೈಸ್ ಆರ್ ಯೂಸ್ ಟು ಟೆಸ್ಟ್ ದ ಸೇಫ್ಟಿ ಆಫ್ ಪೋಲಿಯೋ ವ್ಯಾಕ್ಸಿನ್. ಕೆಲವೊಂದು ಮೈಸ್ ಇರ್ಬೋದು, ಕೆಲವೊಂದು ಮಂಕೀಸ್, ರ್ಯಾಟ್ಸ್, ಸೊ ಅದನ್ನ ಏನ್ ಮಾಡ್ತಾರೆ ಟು ಟೆಸ್ಟ್ ದ ವ್ಯಾಕ್ಸಿನ್. ಸೊ ದೇ ಆರ್ ಯೂಸಿಂಗ್ ಫಾರ್ this purpose also. And chemical safety testing means toxicity testing. So ಹಾಗಂದ್ರೆ ಏನು? Some transgenic animals carry genes which make them more sensitive to toxic substances than the non-transgenic animals. They are exposed to the toxic substances and effects are studied, so which gives the immediate results. ಸೊ ಕೆಲವೊಂದು ಆರ್ಗಾನಿಸಮ್ಸ್ ಅಲ್ಲಿ ಕೆಲವೊಂದು ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಎನಿಮಲ್ಸ್ ಅಲ್ಲಿ ಏನ್ ಮಾಡಿರ್ತಾರೆ, ಸೊ ಅವು ಅಲ್ಲಿ ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಆಗಿರೋದ್ರಿಂದ ಸೊ ದೇ ಆರ್ ವೆರಿ ಸೆನ್ಸಿಟಿವ್ ಫಾರ್ ದ ಟಾಕ್ಸಿಕ್ ಸಬ್ಸ್ಟೆನ್ಸಸ್ ದೆನ್ ದ ನಾನ್ ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಅನಿಮಲ್ಸ್. ಸೊ ಈಗ ಸೊ ದೇ ಆರ್ ಎಕ್ಸ್ಪೋಸ್ ಟು ದ ಟಾಕ್ಸಿಕ್ ಸಬ್ಸ್ಟೆನ್ಸಿಗೆ ಎಕ್ಸ್ಪೋಸ್ ಮಾಡಿ ಹೌ ದೇ ಆರ್ ಗೋಯಿಂಗ್ ಟು ರಿಯಾಕ್ಟ್ ಟು ದ್ಯಾಟ್, and the studies are to study the effects of that toxin. So these transgenic animals are used. Okay, chemical safety testing, then vaccine safety, biological ವ್ಯಾಕ್ಸಿನ್ ಸೇಫ್ಟಿಯಲ್ಲಿ ದ ಟ್ರಯಲ್ಸ್ ಫಸ್ಟ್ ಅಲ್ಲಿ ಪೋಲಿಯೋ ವ್ಯಾಕ್ಸಿನ್ ಇರ್ಬೋದು, ಕೊರೋನಾ ವ್ಯಾಕ್ಸಿನ್ ಇರ್ಬೋದು, ಅದನ್ನ ಏನ್ ಮಾಡ್ತಾರೆ, ಫಸ್ಟ್ ಅನಿಮಲ್ಸ್ ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಅನಿಮಲ್ಸ್ ಮೇಲೆ ಟ್ರೈ ಮಾಡ್ತಾರೆ. ಅದಾದ್ಮೇಲೆ ಏನ್ ಮಾಡ್ತಾರೆ, ಸೊ ದೇ ಆರ್ ready for the that is ready for the human beings. Okay, so these all are the benefits of the transgenic animals. Then the last topic, that is the ethical issues. So problems, so that is related to the biotechnology. Okay, so genetic modification. So that may cause unpredictable results when the organisms are introduced into the ecosystem. So for example, we have introduced some plants or some animals, some particular genes into that animals, and when we introduce that animals into the ecosystem, unpredictable results ಕೆಲವೊಂದ್ಸಲ ನಾವು ಇಮ್ಯಾಜಿನ್ ಮಾಡಿರೋಕ್ಕಿಂತ ಬೇರೆ ಯಾವುದೋ ಒಂದು ರೀತಿಯಾದಂಥ ರಿಸಲ್ಟ್ಸ್ ಬರ್ಬೋದು. ಸೊ ಇಂಡಿಯನ್ ಗವರ್ಮೆಂಟ್ ಹ್ಯಾಸ್ ಸೆಟ್ ಅಪ್ ಅ ಆರ್ಗನೈಸೇಷನ್, ಸೊ ದಟ್ ಈಸ್ ಎ GEAC, ಜೆನೆಟಿಕ್ ಇಂಜಿನಿಯರಿಂಗ್ ಅಪ್ರೂವಲ್ ಕಮಿಟಿ. ಸೊ where the genetically modified research and safety of genetically modified organisms, so that is that will make the decisions. So genetically modified plants irbodu, animals irbodu, directly that is not introduced into the society or the ecosystem. So first the effects of that genetically modified organisms are uh, tested, and after that only, after the safety test only, the genetically modified organisms are used for the public service. So this approval is required. So then the very important topic, that is the biopiracy. What is this biopiracy? So usage of the some resources without permission. Okay, so here biopiracy means usage of the some biological resources without permission, proper authorization. Okay, so that is called as the biopiracy. Then so it is the use of bioresources by multinational companies and other organizations without proper authorization from the countries and the people concerned. So for some biological products, they have some authentication or some authorization for that product. But some multinational companies using that products without proper authorization, that is called as the biopiracy. Certain companies got patents for the products and technologies that make use of genetic materials, plants, etc., that have been identified, developed and used by the farmers and indigenous people of a country. So for the particular product, for example, some herbal medicines like turmeric, neem and all, so India have the patent, okay, to use this herbal medicines or to grow this in a large manner. So in the other countries, they have to take the permission from the patents, the organization or the companies who have the patents, they should give the permission to grow that. Okay, so but without permission or without any proper patents permission, so if they are growing or using that material means, so that is called as the biopiracy. So here basmati rice and herbal medicines like turmeric, neem, so these all are having the some patents. Okay, so you know that basmati, you know about the basmati rice that has the unique aroma and the flavor. So in India around 27 varieties of basmati rice is there. Okay, so genetically different species. So in the year 1997, an American company got patent rights on the basmati rice. So, but it is originated in India. But the patent that was taken by the US based company, that is, this allowed the company to sell a new variety of basmati. So the companies, they are selling the new varieties of basmatis, but that new variety that is developed from the Indian farmers varieties. So that is Indian basmati was crossed with semi dwarf varieties and claimed as the novelty. Other people selling basmati rice could be restricted by patent. ಸೊ ಇಲ್ಲಿ ಏನಾಯ್ತು, ಇಂಡಿಯನ್ ಕಂಪ್ನೀಸ್ ಏನ್ ಮಾಡಲಿಲ್ಲ, ಪೇಟೆಂಟ್ ಅನ್ನ ಬಾಸ್ಮತಿ ರೈಸ್ ಪೇಟೆಂಟ್ ಅನ್ನ ತೆಗೆದುಕೊಂಡಿರ್ಲಿಲ್ಲ. ಕಾಮನ್ಲಿ ಬಾಸ್ಮತಿ ರೈಸ್ ದಟ್ ಈಸ್ ಗ್ರೋನ್ ಇನ್ ಇಂಡಿಯಾ ಓನ್ಲಿ, ಇಂಡಿಯಾದಲ್ಲಿ ಮಾತ್ರ ಬೆಳಿಲಾಗ್ತಿತ್ತು. ಬಟ್ ದ ಮಲ್ಟಿನ್ಯಾಷನಲ್ ಕಂಪ್ನಿ ದಟ್ ಟುಕ್ ದ ಪೇಟೆಂಟ್ ಟು ಗ್ರೋ ದಿಸ್ ಬಾಸ್ಮತಿ ರೈಸ್ ಬಿಕಾಸ್ ಇಟ್ ಡೆವಲಪ್ ದ ನ್ಯೂ ವೆರೈಟಿಯನ್ನ ಡೆವಲಪ್ ಮಾಡಿತ್ತು. ಬಟ್ ದಟ್ ಈಸ್ ಆಲ್ಸೋ ಬೈ ಕ್ರಾಸಿಂಗ್ ದ ಇಂಡಿಯನ್ ಬಾಸ್ಮತಿ semi-dwarf varieties, but it claimed the novelty and got the patent for the basmati rice. Okay, so that if any companies or any country that is having the patent for the particular product means, so the remaining countries, so if they want to sell or if they want to grow that in a large manner means, they have to take the permission to grow that. Okay, so that is about the patent and the ethical issues. So in this chapter, very importantly, the transgenic animals. So what are the benefits of the transgenic animals? That is very important to study the regulation of genes on normal physiology and development, and the contribution of genes in the development of disease, then biological products and the vaccine safety and chemical safety testing. Then so this ELISA, that is antigen antibody interaction test. So that also a molecular diagnosis for the AIDS disease, and how the uh, biotechnology that is used in the molecular diagnosis. Then gene therapy. So she is the first clinical gene therapy, so which was given for the 4-year-old girl. So then genetically engineered insulin, that is the use of the development of the genetically engineered insulin, how it is produced. Then so that is in a medicinal field. Then in the agricultural field, so nematode resistance plant and the bt cotton. So these all are the very important topics in this chapter. bt cotton, how it is going to be a pest resistance plant, and nematode resistance plants, so that is the application in agriculture. Then application of biotechnology in the field of the medicine, that is gene therapy, then genetically engineered insulin and molecular diagnosis. Then some important transgenic animals, how the transgenic animals are have the benefits. Then about the biopiracy and the bio biopiracy, and the patent, about the patent that we have studied. So these all are very important topics for your board exam as well as CET. Okay, I hope this class is useful for you people. If you have any doubts, you can ask. Okay, thank you.